Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Tarek Odali from Business Review, and I will be your host. It is our pleasure to have Piramal Pharma Solutions with us today, who will be discussing an integrated solution for extractable and leachable components. Today's guest speakers are Dr. Balkrishna Agarwal, Group Leader, Analytical Pharmaceutical R&D, and Mr. Abhishek Shah, Research Associate, Analytical Pharmaceutical R&D. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into that box at the top left-hand corner of your screen and then click submit. We will try to allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer, and that will provide us some feedback. You can do that during or immediately after the webinar. And also, if you click on the green resource list widget, you'll be able to view or download a PDF file that's relevant to this webinar as well. Please use the yellow help widget if you require any technical assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Dr. Balkrishna.
Good morning, good afternoon, and good evening to everyone. I'm Balkrishna Agarwal, and I'm a group leader here in Piramal, and I'm responsible for all acceptable leachable activity in Piramal. So today's webinar is all about integrated solution for acceptable leachable. If everyone is on slide number 1, we are now starting this webinar. To start with this webinar, I would like to have a small introduction. What does acceptable means? So acceptable is a chemical species that can be extracted from a primary container and or packaging component under a stress condition. So this is all about acceptable definition. Coming to leachable. So to understand what is leachable, an extractable that actually migrates into a drug product during a normal storage condition is called leachable. So whenever any organization is performing an acceptable leachable study, they need to understand from where acceptable will come and from how leachable will impact the drug product. So these are the two different things. And on slide number 3, why acceptable and leachable study is required? So the primary objective of having an acceptable leachable study is components within the packaging systems which are part of the manufacturing process may have a safety concern. So the primary systems and the closure have become mandatory requirement to meet the safety requirement for marketed medicine. So any packaging material which is being used for any drug product, the safety of the components which can migrate from the packaging material to the drug product, we need to evaluate that safety threshold limit for that particular component. Also, this area is growing very significantly, where regulatory team is facing a challenges for different dosage forms. And these different dosage forms are inhalation, ophthalmic, and injectable drug product, where acceptable leachable study is becoming mandatory. In order to obtain successful regulatory approval for any new drug application, packaging interaction with the drug product should be evaluated to ensure product safety. So to have a foolproof drug product in a US or Europe market, extractable leachable plays a vital role, a vital study to be part of your submission document. That's the reason why acceptable leachable study is required, and nowadays it is mandatory for the drug product which we need to file in these markets. So coming again extractable compounds that can be extracted from the container closure system when in presence of solvent. So during the extractable study, as a scientist, we deliberately try to extract all the chemical components which are present in the packaging material by exposing it in a different solvent medium. The purpose of this extractable study is to screen and monitor presence of toxic material which are the part of your packaging material. And for example, these components can be nitrosamine, phthalates. So these are the components which are being used during the manufacturing of the packaging material. Also, the purpose of extractable leachable study is to develop an analytical method for leachable study, because extractable study is a one-time activity, whereas leachable is throughout the shelf life. We need to conduct the leachable study at different time point when the samples are incubated. Also, the purpose of extractable study is the determination of analytical evaluation threshold from the safety concern threshold. So these are the two terminology which are being used frequently in extractable leachable study. The abbreviation which generally the industry use is AET and the SCT. So to conduct any extractable leachable study, it is very important to identify what is your analytical evaluation threshold and what is the safety concern threshold for the individual component which will be extracted during the time of extractable study. When we do the leachable study, so the compounds that leach into the drug product formulation from the container closure as a result of direct contact with the formulation. So we can think about, if you have one packaging material that can be used for different dosage form, so whether your extractable and leachable will have the same data. So that is the question which needs to be raised in the mind of the executor who are conducting this kind of study. So the leachable study for every drug product is different. So once you conduct extractable study for any primary packaging material, you need to have a full-fledged leachable study. Leachable study is mainly done in the presence of your finished product. And the purpose of having a leachable study is to monitor and control container closure system derive impurities in the drug product during the stability. So the leachable which are part of your drug product during your shelf life, it can be the concern because the impurity levels for your drug product can exceed the specification which is being given in the guidelines. Also, the mandatory materials from labeling, secondary packaging material, reaction product between formulation and container closure system. These are also the critical pathway where your drug product can get contaminated with these leachables. Coming to a slide 5. What is the difference between extractable, leachable, and categories? So the extractable may be different than leachable because different extracting condition, we can get a different components which can come out from the packaging material. So your extraction plays very important role. Secondly, the time frame. The time frame for the extraction, how long you can extract, because this is a continuous dynamic process. You go on extracting and you will find n numbers of extractables. So you need to have that kind of understanding when to stop this. So the time frame plays very important role in extractable leachable study. Also, particularly extractable or leachable can occur in more than one components of container closure system. So there is possibility where different components are present in a same packaging container closure system. So this is all about acceptable. Now, what are the different categories of acceptable which can be present into the packaging system? So the different categories of components are nitrosamine, phthalates, different elements. There are volatile impurities, there are semi-volatile impurities. Then the next category is polynuclear aromatic compounds and antioxidants. So these are the various categories of acceptable which can come out in your drug product when you start doing the leachable study. And all these categories are mentioned in the PQRI guidelines. They have a stringent limit for these kind of components. If these components are present, they have given guidelines where we need to follow, and as per the dose calculation, we need to quantify all these components. Coming to the slide 6, the needs and importance of acceptable leachable study. If you see, USFDA nowadays, they have a concern where they are saying drug and cosmetic, they have given a section where they say a drug is deemed to be adulterated if its container is composed in whole or part of any poisonous or defectious substance which may render the content injurious to health. So FDA has this as a section where they are clearly mentioning container closure is one of the integral part which needs to be evaluated for any drug product. Also, there are biologics 21 CFR 600 where they mention all final container and closure shall be made of material that will not hasten the deterioration of the product. So these are the different guidelines where EMA is also very concerned about the packaging material. The EME guidelines, they say the aim of acceptance study is to determine those additives such as antioxidant, plasticizers, catalysts. These are the things which can be extracted by the active substance in the contact with the plastic material. So the acceptance studies are considered necessarily for plastic material used for container systems of non-solid active substance and non-solid dosage form. So very clearly EMA is saying product which is in liquid form, it can be parenteral, it can be ophthalmic, it can be nasal drop. So all this drug product needs to go through the acceptable and leachable study. In a continuation, there is a Schedule M. Schedule M says all container and closure intended for use shall comply with pharmacopoeia and other specified requirements. There are requirements from pharmacopoeia where they say all these toxic components needs to be quantified, and also we need to determine the purity of the drug. FDA guidelines for industry container closure systems for packaging humans and biologics says packaging components should be constructed of material that will not leach harmful or undesirable amount of substance to which a patient will be exposed when being treated with the drug product. These applications should contain an extraction study on the packaging component to determine which chemical and or residue may migrate into the dosage form, and a toxicology evaluation of these substances needs to be conducted. So these are the different guidelines where they have come across, and they are making it very sensitized. These kind of drug product needs to have acceptable and leachable study. Coming to the next slide, FDA has given a risk analysis on the basis of the degree of concern associated with the route of administration. So just to sensitize the different dosage form, the different route will have a different degree of concern. In this particular slide, there are three different categories for the route of administration. The lowest is low, then high, and then highest. And the packaging component degree is also categorized into three categories: high, medium, and low. So when we see this table very carefully, we can see inhalation, aerosol, and solution are in the highest category of degree of concern when it is administered. And also the packaging material interaction is very high. In the same category, injection and injectable suspensions are also at the topmost concern, where the degree of administration and the dosage packaging components are at very highest level. So the injectable and inhalation product are at very highest level, whereas on the same slide you can say there is a category called low and low. Low for the degree of route of administration and the degree of the interaction with the packaging component. So the lowest is the oral tablet and the capsule. So for oral tablet and the capsule, the degree of concern is very low. So this is how the FDA has categorized the different dosage forms, for which dosage form extractable leachable is mandatory, and for which dosage form the degree of concern is low. It does not mean that the solid and the capsule drug dosage form should not have a extractable leachable study, but it is purely designed on the case-to-case basis with respect to the drug product and the packaging component which has been used. So the complete justification and the protocol are being defined for the solid dosage form. But primarily the inhalation and the injectable, it is mandatory to have such kind of protocol well in advance once you have identified your packaging material. Before you go to the GMP site, my recommendation would be we need to have a extractable leachable study along with the final packaging material which will go into the commercial site for the drug product. So that is my recommendation for extractable leachable study. Coming to the slide 9, I will hand over to Mr. Abhishek Shah, who is one of my associate, and he is doing day in and out extractable leachable study. So I will hand over to Abhishek Shah. Abhishek Shah, please, you can continue from this slide.
Hello everyone. I hope Dr. Balkrishna Agrawal has made clear everyone with what is extractable leachables, why is it required, and what is the basic difference between the extractable leachables, and why what is the need and importance of doing extractable and leachables. So now I'll be guiding you through what are the guidelines available to carry out extractable leachable study. What are the factors one should consider while carrying out the extractable leachable study, and what is the support and service as a being Piramal we are giving for extractable leachables. So continuing the same topic, need and importance of the extractable leachables.
Hello everyone, we seem to be having some technical problems. We'll be right back with you as soon as we can. Audience members, if you would like to interact with some of the widgets that we have on offer while we wait for the speakers to reconnect. And also, please submit your questions for the Q&A session at the end by using the questions and answers widget at the top left-hand side of the screen.
Hello. Yeah, sorry for the interruption. So continue on the same topic, what is the need and importance of doing ENL study? Like these are some typical examples of warning letters which was issued by FDA to some of the industry. Like one of the industry failed to do extractable leachables on the filters. Like one of the company has not done extractable leachables when they were using stoppers for an allergic product. So FDA has issued some of the warning letters to companies for not doing extractable leachables. So there is a need of doing an extractable leachable study. For the same, they have provided a quality control approach which says that one needs to characterize or identify all possible extractables and establish a profile for each packaging component. Like you don't need to characterize each and every impurity coming as extractables. You can identify all possible extractables. The one should establish a correlation between extractable and leachables. Like the extractable which are crossing the AET limits, one should identify that which are those extractables, and you need to correlate it with the leachables. After that, after identifying the potential extractable, one need to set the meaningful extraction criteria with respect to the packaging component based on qualification level and actual observed data. So you need to monitor all those impurities which are crossing the AET limit in your leachable studies. So to carry out ENL studies, there are guidelines available. The guidelines are PQRI guidelines, PODP guidelines, and there are USP chapters available. So PQRI, known as Product Quality and Research Institute, which has given a guideline how to carry out particular extractable and leachable studies. PQR has also issued one of the guidelines which is called as PODP guideline, which is for to carry out the extractable leachable study for parenteral and ophthalmic drug products. There are also 3 USP chapters available to carry out the same activity. The chapters are 1663, 1664, and 1665. So we can refer these guidelines to carry out the ENL studies. So this is just a typical example, like how is the extractable leachable study should be carried out. So first thing is you need to do the extractable study. After doing the extraction study on the packaging component, you should optimize the method. After optimizing your method, one needs to go for the validation, followed by the leachable study. Once you validate your method, then only you can go for the leachable study. And in leachable study, if any component is crossing AET level, you need to go for the toxicological evaluation. So this is how extractable and leachable study works out. So now let us have a look what are the sources of extractable and leachables in our packaging material. So in a pharma industry, when one receives a packaging container or a component, that packaging material has undergone through many industries, starting from monomer synthesis to molding shop. Now these manufacturers tend to add chemical stabilizers, antioxidants, vulcanizing agents, lubricants, colorants, and at the end, when we receive the packaging material, it contains all these kind of components which can come as leachables in our formulation samples. So continue on the same topic. This is a typical example of packaging material what we use in pharma industry, say for rubber stopper, PVDP syringes, like plastic components, rubbers, inks, adhesive forms. These all contains vinyl monomers, vulcanizing agents, antioxidants, organic oligomers. So these all components can come as a leachables in the formulation. So starting with the extraction of packaging material. So when we receive the packaging material, we should start with the extraction study. So this is the example like how we can start the extraction of packaging material. So extraction can be done using high temperature and solvent extraction. So you can use different polarity solvents starting from polar to non-polar, and using the higher temperature, you can do the extraction study on your packaging component. For an example, you can use Sox-Dash extraction. You can directly heat your packaging component, or you can use the reflex process to extract all the components from your packaging components. So when you are doing the extractable study, there are certain factors which you need to consider. Therefore, when you start with the extraction study, you should be aware that what is the primary packaging component, what vendor has provided you. So you need to look out for the material of construction of the system, surface treatments, and the processing aids which are used in the manufacturing of the container closure system. So once you have an idea that these are the components which are used in the packaging material component, it will be easier for us when you are doing the extraction study. So once you can request our vendor that whatever kind of material they have used in the manufacturing process, they can provide us the same. So we are able to quantify the same. So we can identify that potential. We can identify potential extractable leachable with that is associated from vendors, and you can do the literature search. From the MSDS information, you can get the information whether the compound is toxic or not. And more on this, Dr. Balkrishna Agrawal can add his experience also.
Hello everyone, I am back on the webinar. So just to add few more information on this particular slide, where we are on slide number 16. The evaluation of this packaging component plays very important role. Anyone can ask, if you are having a packaging material from a same vendor, I can say if a rubber stopper is being procured from vendor X, so that vendor X will have that acceptable information for that particular rubber stopper in the certificate of analysis. So when we conduct any acceptable study, it is very important to understand whether vendor is providing all this detail. If not, then as a owner of that product, where we are using the rubber stopper from that particular vendor, we need to ask, we need the certificate of analysis where the acceptable information is present in the certificate of analysis. So if you have a rubber stopper from that particular vendor, so we need to do the acceptable study again with our drug product. Many companies, they don't understand why acceptable study needs to be repeated again if vendor is providing the information in the certificate of analysis. Just to update, the acceptable study is for your drug product, not for the rubber stopper. So as a finished product, whether you have aqueous drug product or whether you have non-aqueous drug product, the behavior of your drug product can have a different acceptable in your drug product, because the migration from the rubber stopper to your drug product purely depends on the nature of the drug product. So yes, your vendor will give all the detail of acceptable. Say they have provided 10 acceptable in the certificate of analysis. There is a possibility your drug product is not capable to extract all those components. So yes, during acceptable study, we forcefully do it, and we try to see whether this acceptables are able to evaluate by the technique which we'll be using during leachable study. So for all the drug products, we need to have this acceptable study done, irrespective of the same rubber stopper is being used for all different drug products. So this is how we try to guide all our clients when they come us with all their queries. So these are the potential things when we try to evaluate. We try to give all this kind of solution to all our clients. So coming to the slide 16, acceptable studies with solvent representative of the drug product. Perform acceptable study with the solvent representative of the drug product. What does it mean? The same conversation what I was having. Suppose your drug product is non-aqueous, and if you have a few percentage of ethanol in your drug product, So you need to perform the study with your rubber stopper in presence of ethanol. There might be other non-aqueous solvent present in your drug product. So acceptance study is mainly dependent on what is present in your drug product. It is not about the rubber stopper. It is about your drug product. What is inside your drug product? What is the vehicle? What is the medium which you are using? Again, you need to have a stress condition for doing the acceptable study. So your solvent, your temperature, and your time plays very important role. Just to give you an update, if your product shelf life is at, your temperature is at 25 degrees, and if you do your extraction at 80 degrees, 90 degrees, you are bound to get all these impurities. But whether it is required, it's like you are doing the forced degradation study for your drug product. So the time, solvent, and the temperatures plays very important role. Once you perform this stress condition study, the acceptable profile is acceptable for the specific dosage form, and the level observed will not be appropriate or exceeded during the shelf life of the drug product. So you need to be very sensitive. The levels which you are getting, whether these are the appropriate level, the techniques which you have employed to conduct your study, whether this is a sensitive technique or whether it is a right technique. So all these things plays very important role while performing an acceptable study. Coming to a leachable study. Leachable study in a layman language, it's purely a stability study for any drug product. The same way when we do any stability study for our drug product throughout the shelf life, leachable study is the similar activity where at different time point you test your drug product whether anything from your packaging material has leached out. So during leachable study, the test method for the selected potential leachables for stability studies needs to be developed. So that leachable method is developed when you do the acceptable study. Also, you need to have a different types of technique, whether you need to have identification for that particular impurity, whether it is a qualitative or whether it is quantitative. So you need to identify, you need to quantify. Also, you need to have the safety, access of safety of that particular leachable. So to do this, there are different techniques where you can do the safety assessment. Safety assessment can be done by the toxicologists, or there is a software where we run through the software, which is a direct software. Direct software is one of the well-known software. Once you have identified the component, you can run through the direct software and you can access the safety of any leachable. Also, a container closure system found acceptable for one drug product cannot be assumed to be appropriate for another. This is one of the important point on this particular slide. We all need to understand, if as a company we have selected one packaging component for one product, it does not mean if you have performed that packaging container closure system acceptable leachable study, that report can be submitted for all your different drug products. It won't be accepted, because for all your drug products for the same packaging material, you need to again perform an acceptable and leachable study. Hope I am making myself very clear. Acceptable leachable study for the drug product, for the individual drug product, needs to be carried out, and you need to have an independent report for that particular drug product. Coming to the slide 18, as I mentioned earlier during my discussion, what analytical technique you will be adopting. So you need to be very careful in selecting the analytical technique, because nowadays the requirement from regulatory bodies are at ppb level. Gone those days where people used to report in ppm. So you need to have a technique where the sensitivity plays very important role. You need to have an understanding for that particular instrument. You need to have a capability to develop a method by using all these different techniques. So the techniques which are being used is GC-MSMS. So GC-MSMS is one of the instruments where you try to identify all volatile, non-volatile impurities in your compound. Example, all your hydrocarbon, phthalates, pentenes, etc. are the critical components which can be identified by using GC-MSMS. In Piramal, we are one of the experts who are having this machine. Also, we have the machine, also we have a capability, and also we have the understanding how to interpret the data once the data is out. Because once the chromatograms, once the samples are run, and once you see the raw data, it's not so easy to interpret what all things are coming out. There are a lot many evaluations which needs to be considered. There are a lot many things when you see the data, you need to understand. You need to understand all the fragments which are coming, whether it is the MS which you have used, or if there is a requirement, you can go to the MS technique also. There is a LC-MS technique which needs to be used for all your non-volatile organic impurities. These impurities are nitrosamine, fatty acid, etc. So these components needs to be identified by using LC-MS. All the elements which are present in your component, it's a mandatory now. You need to have all the elements to be evaluated along with your packaging material. So ICP-MS is one of the technique where you use for all class 1 elements. So in USP or in EP, you have all the different classes of the element which needs to be evaluated. So that needs to be evaluated for your packaging material itself. So coming to the slide 19, the toxicological assessment and the AET calculation. The AET is analytical evaluation threshold, the abbreviation which we are using here. So you can see this table is being given in the PQRI guidelines. Anyone can open this and can go back and see. These are the concentration and the threshold which is being given. And these are being categorized as a low toxicity, moderate toxicity, marked toxicity, and genotoxicant. If you see, the low toxicity is having a threshold of 150 microgram per day. That is very high limit which has been given. But in a same table, we sensitize the toxicity threshold for genotoxic impurity is 0.15. So these are the earlier guidelines which were presented. So these were very stringent limit for the genotoxic impurities. But now you can see there is a current PQRI classification of PODP. PODP is parental, ophthalmic, and drug product. So for parental and ophthalmic drug product, you have different guidelines, and now there is a current threshold. You see in the above table, the genotoxic limit was 0.15. But the PQRI scientists, they have undergone several evaluations, and they have evaluated n numbers of components, and now they have come up with this new threshold for genotoxic. Depending on the safety, concentration, and threshold, the value is being increased now. So you have a relaxed limit now, and the limit is 1.5 microgram per day. So now any components or any method which you are developing, your method should be very sensitized at this particular level, so that if there is any component which comes at this level, you can identify and you can quantify. And once you quantify, you can go to the direct software and you can do the toxicology evaluation. Getting a primary data from direct software, then you can give it to the toxicologist. So we have all the experts present in Piramal. So what we do is we run through the direct software, and then all the data is being evaluated by the toxicologist. So coming to the slide 19, here we have given the calculation for analytical evaluation threshold calculation. This calculation is from PQRA guidelines, where it is given the dose, the dose mentioned is 1.5 gram divided by the day. So this is very simple calculation which is present, and for the reference we have given one example also. So the 1.5 microgram we have considered the daily dose, the maximum limit for the genotoxic limit. This can be changed from 1.5 to 150 microgram also. But during the analytical evaluation, your limits should be stringent, because if you have a relaxed limit, your sensitivity of your technique plays very important role. So at current stage, during development, you need to follow this calculation, because this will give you the stringent ppb level. So coming to the next slide, this is an evaluation and the impact of extractable and leachable on your product quality. So the quality plays very important role. So the physical observation, what you can see in your drug product can change the color. It can also have an odor. There can be turbidity in your drug solution. If you are having an injectable drug product, your pH can vary, and you will think why the pH is changing. There might be preservative present in your drug product, and you can see the absorption. Also, you can see there is a decrease in the drug potency, or there is an increase in total impurity. And if you have not evaluated the extractable leachable study, you will be thinking what is happening with your drug product. So these are the physical things which you can immediately observe, and you can see if there is any change. Coming to the slide 22. So what we, as a Piramal, what we can provide. So our mission in Piramal is quality on time. We crave for the argument, the human knowledge for evaluating the solving challenges for developing extractable leachable. So in Piramal, we have all the expertise, we have all the techniques. So the development, what we can do is, we can do the packaging materials certificate of analysis evaluation. We can do the solvent extraction study, varying from polarity. Also, the screening and identification can be done by using different techniques like LC-MS-MS and GC-MS-MS. Also, we can qualify the extractable as per the vendor, which is the certificate of analysis, or as per the PQRA guidelines, or as per the USP guidelines. So we can have all this kind of protocol, and accordingly we can do all the extractable study. Also, the validation and the verification for all the analytical methods can be done. Toxicological evaluation as per the direct software can be done, and also the conducting of leachable study. Leachable study is a full-fledged shelf-life study that can be done by Piramal team. So that is what we are offering. Services, support and services. So the research capability what we have here, we have listed down the potential extractable and leachable. So these are the phthalates, nitrosamine, elements, volatile, semi-volatile organic components, polynuclear aromatic compounds, antioxidant and elastomers. So that scientists of Piramal reveals their knowledge and expertise in developing all these components by using various techniques. Also, we have a niche library, the latest library for the GCMS, where there are lakhs of components, millions of components which can be screened, and we can say screen it. Here at slide number 24, we have represented a case study where we have done the MS study. So there is a scan mode. The first chromatogram shows the scan mode where you can see the multiple components which are being presented. Also, there are different modes, the SIM mode and the MRM mode. These are the techniques, these are the terminology which is being used. So when you adopt with the different mode, you can see the different response. In MRM mode, when you inject your sample, the same fragment can be further fragmented and you can have a fingerprint for that particular compound. So you have a unique fingerprinting for that particular component, and that can be achieved by MRM mode. So the laboratory capability. So here you can see this is our R&D site, and the equipments what we are having is GC MSMS, GC LC MSMS. We have all the HPLC with all the detectors. We have GC, we have stability chambers as per the ICH guidelines, and the chambers are 24/7. We have a power backup. So finally, I will like to thank you, all the participants who have joined this webinar, and hope this webinar was helpful and at least it has given some kind of insight to all the attendees who have attended this webinar. Over to Tariq.
Thank you very much. Just a reminder for the audience, we're about to go into our Q&A session now. So you can send in your questions and we will try to get to as many of them as we can. So you can send those in via the questions widget, and that means to just type them into that box at the top left-hand corner of the screen and then click Submit. Okay, so we're going to go ahead and see some of these questions that have been coming in already, but please go ahead and continue to ask them, audience members. First question is, which solvents can be used as extraction study? And
Yeah,
that's
so the answer
directed.
of your question, yeah, so the answer for your question is you can use the different polarity solvents. Preferably, you can use DCM, that is dichloromethane. You can use hexane, heptane, and you can adjust the pH of the water starting from 0 to 12, and like this varying polarity starting from polar to non-polar. You can use any. You can choose any 3 solvents. You can choose this for your extraction study.
Okay, thank you very much for that answer. And okay, here we go. So the next question is, which category of compounds are considered as highly concerned extractables?
So there are compounds which contains nitrosamine groups in their structures. There are compounds which contains polyaromatic hydrocarbons compounds. These compounds can be considered as the highly concerned extractables.
Okay, lovely. Now, moving on to the next question. But before we do, I just wanted to remind the audience you can still ask your questions and do those via the questions widget. So just type them into the box at the top left-hand corner of the screen, then hit submit. So here's the question for you. Does pH of the product play any role in leachables?
Yes, the pH plays a very critical role in the leachables. When the pH shifts from the actual value during its shelf life, there are possibilities of leachables that can be leached out into your formulation during its shelf life if pH shifts from the actual value. So yes, it plays the important role in the leachable studies.
All right, thank you very much. I hope that answered your question. And the next one is it necessary to quantify each extractable?
No, it is not required to quantify all the acceptable, because we have this AET limit which give us the limit. Below that there is no requirement. You can just identify, and you can just putting that identification, you can say this is the component. So all the acceptable did not need to be quantified.
All right, lovely. And here's quite a long question now. But before I go through, I just wanted to remind the audience you can still ask your questions and get them in via the questions widget. Okay, here's the question. So If the same kind of rubber stoppers is being used for many formulations, is it necessary to perform extractable and leachable for each?
Yes, during my entire discussion, I was mentioning, and this is one of the key points. If you have a packaging material for one drug product, and that drug product is aqueous, and now with the same packaging material, you need to have you want this packaging material for a different drug product. Definitely, you need to have a different report, and you need to conduct a full-fledged study for that extractable leachable, because your drug product plays very important role in extractable and leachable study.
Okay, thank you very much for those answers, and I'm sorry to anyone whose question we didn't have time for. We are out of time for the Q&A session now, so I will forward those questions on to the sponsor, and then you'll be able to get a response back from them soon. So that just leaves me to thank Dr. Balkrishna and Mr. Abhishek for the great presentation. I'm sure you all agree, and to thank Piramal Pharma Solutions for sponsoring this session. To the attendees, you'll receive an email very shortly, and that will tell you how you can access the on-demand version of this webinar. You can watch it all over again, or you can access it through our website directly, and that's www.business-review-webinars.com. And once this webinar ends, today's survey will appear in its place, and we would very much appreciate it if you could answer the questions in there for feedback. Also, add directly to the resource list, where there is a PDF file available that might be of interest to you. And we look forward to sharing further webinars with you. Of course we do. So please do keep an eye out on the website I just mentioned. That's www.business-review-webinars.com. And you can follow us on Twitter at BRWebinars for daily updates. And you can also join our LinkedIn group, and that group for you is Business Review Webinars. To the speakers, is there anything you'd like to say to the audience before we end?
So the only message which I would like to share is, yes, thank you very much for attending this webinar, and do connect with us, whether you have a full-fledged project or whether you don't have. We can provide the solution as an expert. We are always there to support any acceptable and leachable study if near future you have any plan to conduct this. And also the questions which are unanswered, we will make sure we will answer each question, and we will answer this via email.
Okay, thank you very much. And thank you to everyone who attended, and thank you for your attention today. And I hope you all have a lovely rest of your day.